In this video, I'd like to continue our discussion of experimental design and analysis in ecology. In the last video, we discussed the different approaches that ecologists use, so observational studies, manipulative experiments, and modeling. When doing any of these things, it's really important to consider the design of your study, how you're going to collect data, how you're going to set up any kind of sampling. So this is a picture of a common garden. So common gardens are really popular ways of doing both observational studies and manipulative experiments in ecology, especially manipulative experiments. So here we see we have a study area and it's divided into plots. Those squares are different plots with different plants planted in them. And you can manipulate all sorts of different things in common gardens, right? So you can change the assemblages of plants. You can put plots with high diversity, low diversity, or somewhere in between. So different numbers of plant species. You can plant different genotypes of plants or different varieties. You can exclude predators or herbivores by putting netting or something over the plots. You can change the soil nutrients. You can add nutrients to some plots and not others. And where this is powerful is you can basically set up treatments and controls, or different treatments. You can set up high, medium, low, or different levels of any kind of treatment that you devise. But what's important in a common garden is you have to replicate, right? So if you have different plots where you added nitrogen, for instance, you need to have multiple replicates, and you have to have multiple replicates of your control. And then you also have to randomize. So you have to randomly place those plots within the common garden. So if we think about why do we randomize and why do we replicate? In the last video, we talked about correlation is not causation. And we said, well, X could cause Y, or X and Y could be caused by Z. So if we see variability in X or Y, so our independent or dependent variables, they might be caused by some unmeasured variable. So we randomize and we replicate mostly to account for unmeasured variability. So in this common garden, we can manipulate something. We can manipulate, say, soil nitrogen, but there may be other things that vary as well. You can see that some of these plots are in the shadow at this time of day and some are in the sunlight, so there may be different levels of light. There may be different soil textures that hold different amounts of moisture. There may be different sorts of effects from the edges of the garden, right? Different plant species or different animals or so on, right? So these are things that aren't explicitly measured in the manipulative experiment, but we have to account for them. And we do that by having lots of samples, lots of treatments, lots of controls, and we can average out the effects of unmeasured variability as long as the plots are properly replicated and properly randomized. So there's a right way and a wrong way to randomize. And if you do it the wrong way, we run into a problem called pseudoreplication. Basically, we think we have more replicates. We think our sample size is bigger than it is. So consider this example. We want to figure out the effect of fire on plant species diversity, right? So we have one area in red here where we had a burn. We either experimentally burned a field or it was a wildfire. And then we have site 2 that's unburned. And then we can put plots in here. And the plots are the yellow squares. And so we might say we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 plots in the burned, in the treatment, and 10 plots over here in the control. So you might think, well, our n is 10, right? Because we have 10 replicates. And so if you calculate your standard errors, your standard error bars would be relatively narrow, depending on how many, what your sample size is, but we'd be wrong. We really don't have 10 replicates here because if you look at this, we don't really have any randomization or replication. This is one burned area and this is one unburned area. And there could be unmeasured variability between them. The two, they could have different soil types. They could have different plant assemblages. They could have different herbivore pressure, right? We haven't really accounted for unmeasured variability. And so we have a pseudo-replication problem. If we set our n as 10, our standard errors would be narrower than they really should be. The n should be 1. and so our standard error should be really wide to see if there's a true difference between burned and unburned. So the way to do this correctly and avoid pseudoreplication is you have to have a large enough study area where you're going to have multiple burned and unburned areas. So here we see we have unburned plots as yellow and burned plots as red. They're dispersed across this area and they're randomized. So now we truly have 10 burned and 10 unburned samples. And so now our n is 10. We can calculate our standard errors based on that. So when designing your studies, designing your experiments, whether it's observational or manipulative, you need to make sure that you avoid pseudoreplication. Okay, one other thing I want to mention in this video is something that we're going to do for this week's lab. So you have your you have your leaf litter invertebrates, and you're going to calculate a rank, or you're going to make a rank abundance plot. This is sometimes called a Whittaker plot. And what a Whittaker plot is, we rank the species by abundance. So on the x-axis here, it's you put the most abundant species as rank 1, and the least abundant species farther down the x-axis here. And then on the y-axis is the proportion of abundance. So for each species, how many individuals do you have? And how many individuals of that species, what's the ratio of the number of individuals of that species to the total number of individuals of all species in the sample. And what a rank abundance plot tells you is not only the number of species, but also the relative abundance of the different species. So over here we can see two definitions. So species richness is the total number of species present in an ecological community or in a sample. So we take a sample of leaf litter invertebrates and we can identify all the species, and that the total number of species is the species richness. Right? So we could compare two different places and we can compare species richness, but that's an imperfect measure of diversity because that richness could be distributed very evenly across species, or it could be dominated by one species. So species evenness here is the measure of how close the abundance of different species are to each other. So for instance, the black line here on our plot, the Centennial Brook, you can see that's a very steep rank abundance curve. So we have a lot of species 1, and then we drop off very steeply, right? So the other species are pretty low abundance. If we look at the blue line, Snipe Island Brook, we see a much more gradual rank abundance curve here, and so abundance or dominance is shared among multiple species. So a rank abundance plot is sort of a visualization to be able to compare the diversity between different plots. In another video, I'll show you how to make one of these in R, and you'll put your leaf litter invertebrate data into R and create a rank abundance plot.